All right. Well, good afternoon. Welcome to this briefing from the American Society of Gene and Cell Therapy. I know folks are coming in from the waiting room, so glad to have you. Today we'll be covering next generation cell and gene therapy approaches for HIV. My name is Christina Mayer. I'm ASGCT's Director of Government Affairs, and today we have an excellent lineup of scientists and advocates who will dive into this really critical topic. One housekeeping item for attendees before we get started. There is a Q&A box at the bottom of the Zoom window there, so feel free to submit your questions. You can do so anonymously if you prefer, and then we'll have about 20 minutes at the end held for our panelists to answer those questions. So with that, we can kick things off. We are very grateful that this briefing has been sponsored by Senator Chris Coons of Delaware, who is a member of the bicameral bipartisan HIV/AIDS caucus, which has done some really great work. Senator Coons was kind enough to record a video welcome, so I will hand it off to him.
Hi, I'm U.S. Senator Chris Coons from Delaware. As a young man, I worked with a program serving orphans in Kenya. It was in the early 80s, and there was a wave of suffering and death that hadn't yet been fully understood or even named. Locals called it slimming disease. The world would later learn this was HIV/AIDS, and it was spreading everywhere like a cloud over the globe. Now, for the first time since those early days, when the pandemic of HIV/AIDS was identified, a world without HIV is no longer just a dream. From gene therapies to a new injectable pre-exposure prophylaxis for HIV, we're living through a paradigm shift in how we treat and prevent HIV and even possibly cure it. Breakthroughs like these don't come from one person in one lab. They come from collaboration, patient advocates and researchers, industry and policymakers working together. And they come from funding the basic research on which life-saving cures are built. I'm proud my home state of Delaware is part of this work, from the Gene Editing Institute at Christiana Care to startups working at the intersection of AI and cell therapies. It's the kind of life-changing research made possible by decades of federal investments, and why I'll never stop fighting in Congress for the world-class research funded by the NIH. The new technologies we're discussing today could change the lives of more than 1 million Americans living with HIV, and by exporting this American innovation around the world, we could stamp out HIV once and for all. To the researchers and advocates sharing their experiences today, thank you. Because of you, the dark clouds of HIV around our globe are clearing, and we can see a tomorrow where no person, no matter where they live, has to live in fear of HIV. Hi,
So thank you again to Senator Coons for his work in this space. Moving on to our panelists, though, I'd like to first introduce Dr. Paula Cannon, who is ASGCT's immediate past president and a distinguished professor at USC's Keck School of Medicine. Dr. Cannon will kick things off for us, and then we'll introduce each of these 4 expert speakers in their turn. So, Paula.
Thank you, Christina. Hello and welcome to everybody. Yes, so I'm here both as the scientist who works on HIV, but also as a representative of ASGCT, which is co-sponsoring this event. And I'm going to give you a high-level overview of why we think cell and gene therapy is applicable for HIV. Next, please. Thank you. So just as background, ASGCT is the primary professional membership organization for gene and cell therapy. We've got over 6,000 members, and we're scientists, clinicians, and patient advocates. And we come from lots of different backgrounds, and we take on lots of different activities. I'm very proud to be a member of that society. So just starting off with a very simplistic way of thinking about disease. And because I'm a gene and cell therapist, I'm afraid that this is how I view the body and how I view disease. I view the body as just a bag of genes that have the information to produce all the processes necessary for life. And so when disease happens, I think of it as being either, bad genes or bad cells. And those bad genes can include genes that are mutated, that you are born with. Think of things like hemophilia. Or we can acquire bad genes, particularly from viruses. We now know that Epstein-Barr virus can lead people to get MS later in life. And then, of course, individual genes can mutate and drive cancers. And the sorts of bad cells we have in the body include defective, missing, or aging cells, or rogue cells that cause cancer, or even attack our own bodies in autoimmune diseases, things like lupus. Next, please. So this slide shows the basics of cell and gene therapy, and I've divided it into three different buckets. So first of all, the basic idea of gene therapy is that we can repair or add a new gene to a cell to combat the disease. And in some cases, this can be done by directly injecting that new gene into a body using what we call gene therapy vectors, and that's shown at the top. And they enter the body and they find the necessary target cells and they deliver the gene therapy medicine. But we can also directly think about using modified cell therapies as well. And cell therapies can be two types. So first of all, down at the bottom in purple, I'm showing the idea that you can actually replace defective cells in the body with donor cells from a tissue-matched donor. So this would be things like a bone marrow transplant. Or we can also take cells out of a person's body, typically this would be blood cells, and we can genetically modify them in the lab and then return them to the body. And a good example of this, it would be CAR T cells, which are revolutionizing how we treat some cancers. Next, please. So cell and gene therapy offer unique benefits as a type of medicine because they target the underlying cause of disease, the defective gene or the defective cells. And we also imagine that many gene therapies will be a one-time administration with the goal of being curative. And importantly, gene and cell therapies are applicable to the many rare diseases that can be devastating, but for which patients have few or no options. And on the right there, I'm just showing some of the many FDA-approved cell and gene therapies that are now available, and they cover applications from cancer, genetic diseases such as sickle cell, hemophilia, muscular dystrophy, deafness, blindness, and a whole long tail of rare congenital diseases that may only impact a handful of people, but for whom a gene therapy offers the only option for a treatment and a cure. Next. So I'm going to touch on two examples of where I think gene therapy and cell therapy are having an extraordinary effect even now. And the first example is sickle cell disease. Sickle cell disease is a significant unmet need in this country. There are estimated to be 100,000 Americans living with this disease, and 1 out of every 365 Black or African American births leads to a baby who is born with this disease. And excitingly, in December 23, the FDA approved for the first time 2 different gene therapies that are able to treat patients with sickle cell disease. And this has now progressed to a so-called cell and gene therapy access model, which is being overseen by CMS and Administrator Dr. Oz Mehmet, to partner with the states to manage bringing sickle cell disease gene therapy to the many Americans who would benefit from it. Next. The other exciting example I want to just touch on are CAR T cells. I've already mentioned these, and I'm sure many of you have heard of them because they are revolutionizing how we treat certain blood cancers. So in my little cartoon here at the top, I'm showing you what can happen in a cancer patient. We have these cells called CD8 T cells or killer T cells that would normally recognize and kill a cancer cell before it even got started. But in the case of somebody with cancer, one of the problems is that those CD8 T cells are ignoring the cancer cell. So what genetic engineering can do is it can modify those CAR T cells, engineer them so that they can now recognize the cancer cell. And in that way, the CAR T cells can attack and kill the cancer cells. And again, this is being used with leukemias and lymphomas and has been extraordinarily successful. Next. So that was an overview of cell and gene therapy in general, but I want to spend just a couple of slides focusing in particular on why we think this is applicable for HIV. So, of course, HIV is still a massive global and national health problem. In the United States, over 1.2 million people are infected with HIV, and we have over 30,000 new infections every year. And although we have a wonderful medicine cabinet of FDA-approved anti-HIV drugs, which include one-a-day pills that can keep HIV undetectable in many people, unfortunately, the drugs do not cure HIV and so must be taken for life. And also, and this is something that kind of drives me as I think about this, that even though we have these pills, and in theory, they should work very well in everybody, the estimates are that only 50% of people who are infected with HIV actually achieve this long-term HIV suppression due to the fact that, people need to be retained in medical care for the rest of their life to access these drugs. And so because of that, the fact that cell and gene therapies offer the potential for a one-time treatment makes them especially compelling for a chronic lifelong condition like HIV, and also potentially even a cure. So let me just touch on why these drugs don't cure people of HIV. And I think it's for three reasons. HIV really is a triple threat. First of all, this virus has an incredibly high mutation rate. It leaves influenza and COVID and other viruses in the dust. And because it can mutate so quickly, even though we mount an immune response against it, we make things like antibodies. The virus just evolves and mutates and is now resistant to those antibodies. Secondly, HIV actually infects and kills a specific cell in the body called the CD4 T cell. And unfortunately, CD4 T cells are a critical part of our whole immune system. And this is why people with HIV eventually develop a profound immune suppression that can lead to AIDS. And then finally, the virus, as part of its lifestyle, actually inserts its own genetic material into the DNA of an infected cell. And in this way, it creates what we call a reservoir of HIV that is not removed by these antiretroviral drugs. And that means that if people stop taking the drugs for any reason, the virus will usually rebound and sort of flare up again. So this is why the drugs have to be taken for life and they don't cure people. Next, please. So here I'm just showing some examples of different types of cell and gene therapies that are being developed against HIV. In the first case, we now know ways that we can make a person's CD4 T cells, the target of HIV, resistant to the virus. And I'm calling this the CCR5 story, and you're going to hear about it from our next speaker. In addition, we can use new genetic tools called CRISPR that you may have heard of. I think of them as genetic scissors, and they can be used to cut out HIV from infected cells. And then the third type of approaches are improving our natural immune response to HIV. So I've already told you about CAR T cells, which are engineered CD8 T cells that can kill cancer cells, but we can also use them to kill HIV-infected cells instead. And then finally, we can use gene therapy vectors to deliver into the body the cassettes to express potent anti-HIV antibodies better than anything our bodies naturally make that could suppress the virus. And then finally, point number 4, and again, we'll hear about this from our next speaker, we continue to learn new lessons by studying rare individuals who have actually been cured of HIV, and we think about ways we can use that information to develop new therapies. Next, please. So I'll just leave you with a couple of final thoughts. So research and development for HIV has actually taught us many lessons that we now apply to other diseases. There's really been a massive value-added benefit. For example, CAR T cells were first developed in the 1990s for HIV long before they were used for cancer. And then I will also recognize that people living with HIV have been amazing advocates for the type of science and research that we do. And they are willing to participate in many clinical trials that in many cases are first in human trials. So they're very brave, but this has helped to advance the whole field of gene and cell therapy. Okay, and with that, I would like to introduce our next speaker. Our next speaker is Jonas Sasha. He's a professor at Oregon Health and Science Universities and the Oregon National Primate Research Center. And he's going to be talking about lessons from people who have been cured of HIV.
Good morning. Thank you for that introduction, Paula. And it's a real pleasure to speak to you today about the field of HIV research, and specifically HIV cure, because we're standing at a really exciting time where, as I'll talk about momentarily, we have, for the first time, many individuals who have been cured of HIV that are willing to collaborate with us. And I think we're standing on the precipice of being able to actually cure HIV. And it's very likely that those cures are going to be gene and cell therapy based. Next, please. So this story starts with the first individual who was cured of HIV, and that was Timothy Brown, also known as the Berlin patient. He was an American who was living in Berlin at the time, and Timothy unfortunately developed a leukemia, and the treatment for that is a chemotherapy, which unfortunately destroys the stem cells that live within your bone marrow. And those stem cells in your bone marrow give rise to the red blood cells that carry oxygen, and white blood cells that are your immune system in your body. And so in order to overcome that, you require what's called a stem cell transplantation. This is a very dangerous and complicated procedure, as you'll hear about later from one of our next speakers. And the stem cell donor must be tissue matched to the recipient. And this is important because if you don't, the transplanted cells, which contain some of these killer T cells that Paula just talked about, what we call the graft, stem cell graft in this case, will recognize the body as foreign and will attack those tissues of the new host. And those actually can do some very interesting things, as I'll talk about momentarily. But Timothy's donor was really special. His physician did something really unique. Was his donor was not only tissue matched, but also carried 2 copies of a harmless genetic mutation called CCR5 delta 32. And this is a 32 base pair deletion within the CCR5 gene. All that it does is it renders the CCR5 gene unable to be expressed. And so people who have this mutation do not express CCR5. Next slide. And why this is important is because CCR5 is the lock that HIV uses its key to enter CD4 T cells and infect them. So you can imagine if someone does not have CCR5, HIV cannot use its key to get into that lock. It can't infect the cell. Those individuals are markedly resistant to HIV infection. And so these individuals cannot be infected by HIV, and therefore they cannot develop any immunodeficiency or AIDS. And so people who have this CCR5 delta 32, if they have 2 copies of this gene, they don't have CCR5, and HIV cannot infect those cells. And so when Timothy received a transplant from one of these individuals, it meant that Timothy's new immune system, which came from that donor who didn't have CCR5, when it repopulated Timothy's body, also did not have CCR5, and therefore it was HIV resistant, leading to his cure. The virus simply had no place to go because all of these CD4 T cells that it could see didn't have the lock on the door for HIV to get in. Next slide. And this really summarizes where we've come since Timothy. So in 2009, Timothy Ray Brown the case was published, which showed that he was actually cured of HIV. And for the longest time, he was the only individual who was cured. It wasn't clear that this was what the mechanisms were, or if it was possible to cure more people in this way. And unfortunately, Timothy had a recurrence of his leukemia in 2020. But before he died, he was joined in his cure brotherhood by Adam Castillejo, the London patient, in 2019. Timothy actually spoke with Adam and convinced him to release his identity. This was then subsequently followed up in 2022 by the New York patient. This is the first woman who's been cured of HIV. And you'll notice that there's no picture. That's because this individual has decided to remain anonymous. This is an important point because I think it still speaks to that there is a stigma around HIV and thus the need for us to cure it, as well as the discrepancy in health span and lifespan with individuals that are still living with HIV. In 2022, we also were joined by Mark Franke, the Dusseldorf patient, and then our very own Paul Edmonds, who you'll hear from next, the City of Hope patient. And since then, there's been even more cases that have been reported, and there's now a total of 10 individuals who have all been cured by receiving a stem cell transplantation. Next slide, please. So while the CCR5 mutation is incredibly powerful, it is hampered by the fact that it is also incredibly rare. It's less than 1% of individuals in the population have this mutation. And so gene and cell therapy approaches now are trying to copy these cures without the need to use a stem cell transplantation because it's very costly, very difficult procedure. And one of the mechanisms that they're doing this is using CRISPR gene editing. These are the genetic scissors that you just heard about. And what you can use this technology to do is you can take a patient's own stem cells, you can use the CRISPR technology to cut out those base pairs in CCR5, thereby making a patient's own stem cells lack CCR5, and then you can reinfuse them back in almost as if a self-stem cell transplant. Therefore, they would then be resistant to HIV, and this would be a cure for HIV as well as making them resistant to subsequent infection. And so this is an area of very high research, intensive research right now. Next slide. Now, as amazing as CCR5 and as viable as approaches, two of these cases are really unique. These are the ones that are in this box. This is the Geneva patient, which was published last year, and then the second Berlin patient, which actually the case report just came out this week. This is how breaking this is. And both of these individuals were cured by receiving a stem cell transplant, but from a donor who actually has CCR5. So the WT is for wild type CCR5. And this sort of turns on its head this idea of the CCR5 cure, and it opens up new possibilities for cell and gene therapy approaches to cure HIV independent of the CCR5 mutation. Next slide. And so how does this work? Well, it's actually very similar to what happens in leukemia. And so what happens is when you receive a stem cell transplantation from someone, you're receiving not only stem cells, but also some of their immune system cells, specifically some of their killer T cells. And even when that tissue match is great, these donor T cells can still recognize the body that it goes into as foreign and kill it, and attack those cells, sorry. And T cells have one essential function. They look at a cell and they say, is this self? Don't kill, non-self kill. And this is great when those T cells, as shown here on the left side of this fulcrum, when those T cells encounter a tumor cell and they recognize it as non-self and kill it, that's great. That's what we call graft versus leukemia. These T cells recognizing the leukemia cells and killing them. This is actually what cures people of cancer and puts them in remission. Very powerful effect. However, on the flip side of this coin is if those T cells recognize healthy cells, such as your skin cells, as foreign and kill them, they'll mediate what's called graft versus host disease, or GVHD. And this is a very serious complication of transplant. It is actually the most serious complication of transplant. And this is what makes the whole procedure so dangerous. And you'll see the graft versus host disease on the right side of this fulcrum. And one of the major focuses now in leukemia research is to bias that fulcrum over to the left so you mediate this graft versus leukemia while avoiding the graft versus host disease. Next slide, please. And so we actually hypothesize that this is exactly what's happening in these cases of HIV transplant, HIV, where you get a stem cell transplant. We hypothesize that these donor killer T cells in the graft also are clearing the reservoir and therefore mediating cure. And so you'll see on this image, we replace the graft versus leukemia with graft versus HIV reservoir. So instead of a T cell recognizing a leukemia cell as foreign and killing it, it's actually recognizing a CD4 T cell that has HIV inside of it and killing it. And that mediates the graft versus HIV reservoir. And this opens a whole new field of, can we use this to make new gene and cell therapy approaches for HIV cures? Next. And how are we doing this? So here in the upper left, I'm happy to say that my collaborator, Lishoma Ndlovu, here on the left in this image, and myself received an NIH grant to actually study this exact phenomenon and develop it into a gene and cell therapy cure. And why is this exciting right now? Is the picture on the right. Again, on the left is my collaborator, Lishoma Ndlovu, myself on the right. But in the middle, these are the three individuals who have been cured of HIV that have released their identity. It's Mark Franke, the Dusseldorf patient, Adam Castillejo, the London patient, and Paul Edmonds, the City of Hope patient. And this picture was taken the first time they ever met each other. This really was an amazing moment for me, but also for HIV because it showed that for the first time now we have a growing number of individuals that are cured, and we can begin to study them and understand how they were cured to make new, excuse me, cell and gene therapy approaches. And so Mark, Adam, and Paul have generously agreed to collaborate with us. Let us study them, and we're going to study specifically those killer T cells in them that mediated the graft versus HIV reservoir effect and understand what those cells look like. And once we know what those cells are and what they look like, that can then be the blueprint to help us design new cell and gene therapy approaches that allow us to come and target an individual's own immune system to better target their own HIV reservoir and mediate a cure without having to use a stem cell transplantation. Next. And so in conclusion, individuals living with HIV who develop a leukemia do need stem cell transplantation to treat that leukemia. And in these rare cases, this actually leads to HIV cure. As I mentioned, we now have 10 individuals who've been cured of HIV. And this is really remarkable because these cases prove that curing HIV is possible. We can do this. And using these individuals that are cured, that are willing to collaborate with us, we can now study them and understand how HIV was cured. And this could unlock the key to ending HIV for good. And why this matters is because what we learn today can help us build the next generation of cell and gene therapy approaches to cure HIV. And personally, I believe that we will cure HIV in our lifetime with these approaches. Thank you. And with that, we will now move to our interview section of this. And we'll be speaking to Paul Edmonds, the City of Hope patient, and Michael Luella, who is a patient advocate. And so now I'd like to welcome Paul Edmonds. Paul, welcome.
Thank you, Jonah.
So it's a real pleasure to introduce you, Paul. As I just spoke about, you are one of the three individuals who have been cured of HIV who has released their identity. And I would like to say that as a researcher, you are the inspiration that gets me up and out of bed and makes me want to do research every day. So thank you first and foremost. I wanted to ask you though, before you became known as the City of Hope patient, you lived with HIV for 30 years, including a diagnosis of AIDS. Could you please tell me what that was like?
Yes, living with HIV for the first decade was incredibly challenging. In the early years, the stigma was heavy and constant. The medications available back then were harsh, difficult, putting it mildly. And managing side effects became a part of daily life. I lost many friends and acquaintances along the way, which was emotionally devastating. We were also facing a political climate where the administration wouldn't even say the word AIDS. It felt like they wished we would simply disappear. Sadly, some of that attitude echoes today with the current administration refusing even to acknowledge World AIDS Day and cutting HIV funding. Things finally began to shift in the mid-90s when protease inhibitors arrived. For the first time, there was real tangible hope, and life slowly started to stabilize. Treatments kept improving over the years, and I learned how to live with HIV rather than just surviving it. Then 30 years later, acute myeloid leukemia entered the picture, and that's where my story took a very different turn. And as they say, that's history. The rest is history.
It's amazing. Paul, the only other documented case of HIV cure in the United States, the New York patient, has remained anonymous. whereas you have released your identity. Can you tell me what inspired you to release your identity and go public?
Well, I still remember the day I heard that Timothy Ray Brown had been cured of HIV. For the first time in years, I felt a genuine sense of hope that a cure might one day be possible. Timothy's courage helped ignite the field of HIV cure research. Sharing my identity and my story now is my way of carrying that torch forward. I hope it offers comfort, possibility, and resilience to people living with HIV.
Yeah, that's great, and I think it does. So you were cured by a stem cell transplantation. Um, I've been peripherally associated with that, in the transplant wards. But can you tell us what the entire stem cell transplants procedure was like?
Yes, the entire stem cell transplant process was long, exhausting, and incredibly demanding. I spent almost 5 months in the hospital, followed by another 2 months living in a hotel near the medical center. After a transplant, anything can change very suddenly, so you have to remain close to the hospital for constant monitoring. Before I could even begin the transplant process, I went through many rounds of chemotherapy over about 3 months to push the leukemia into remission. Once that happened, I was eligible for the actual transplant. Then came the conditioning chemotherapy, which completely wiped out my immune system to make room for the donor cells. That was, without question, the most difficult phase, physically and emotionally. After the transplant, I did experience graft versus host disease, but thankfully, in my case, was relatively mild compared to what many patients go through. Even so, the risks are always present with a stem cell transplant. Your own cells can fight the donor cells, the donor cells can attack your body. It's a delicate balance. Overall, it was a long and often grueling journey, but one I'm profoundly grateful to have come through.
Thank you. Um, you know, what do you hope for by sharing your story of HIV cure? What do you hope it will achieve?
I hope my story becomes a part of a larger, yeah, I hope that it will become a part of a larger narrative of progress, a reminder that breakthroughs happen because people are willing to step forward and share. If my experience can offer hope today and help pave the way toward a future where HIV cure is accessible to all, then that is the legacy I want to have.
I love it. And just one final question, because I have to ask it, because you were there at the beginning of the HIV pandemic. What would it mean to you to also be there at the end?
Well, it would mean that all those years of fear and grief were not the whole story. It would mean that the courage of the people we lost, the activism of communities, and the persistence of scientists and clinicians led us somewhere towards hope, health, and justice. To be there at the end of the HIV pandemic would feel like seeing history come full circle. It would mean that a diagnosis that once isolated people and marked them for death has become something we can prevent, manage, and ultimately cure. For me personally, it would mean I lived long enough to see the impossible become possible, not just for me, but for millions of others. And it would be a reminder that the decisions you make in rooms like this can help determine whether that ending comes sooner and how many people get to live to see it.
Wonderful. Thank you, Paul.
Thank you.
And Paula?
Yeah. Hi, Jonah. Thank you. So we're going to switch now. I'm going to be interviewing briefly Michael Luella. Hi, Michael.
Hi, Paula.
Michael has worked as an HIV treatment research educator, engaging communities affected by HIV in Seattle and across the US for 26 years now. And he's been especially critical in helping to explain the concepts and the ideas behind cell and gene therapy. So Michael, we talk about an HIV cure. What does that mean to you? And what would a cell or gene therapy actually have to deliver to be truly considered a cure for HIV?
Well, I would say like a cure for most people living with HIV would mean that they no longer have to take pills every day. Um, they wouldn't have to worry about their viral load coming back, and they wouldn't have to think about HIV controlling any part of their life anymore. And so a cure would have to be something that would be safe, long lasting, hopefully forever, and not really leave people with a bigger problem than the virus ever left them with. So it would have to be something that's very safe. So cure for most people, they say freedom. A cure would be freedom, freedom from the virus, freedom from the stigma, freedom from the constant management of HIV. And it's just something that shouldn't work in just a lab. It should make my life better. And that's what most people tell us when they talk about cure.
Right. So that sounds like that as well as having physical benefits, it would have a huge emotional benefit for people.
Definitely. I think that's the part that people say that it would feel like they would get part of their life back. That HIV is very manageable now, but it still lives in your mind, and you think about it like when you're trying to date, or when you're trying to see new doctors, or when you're traveling, when you're trying to just plan for your future. And so, like a cure would feel like a weight being lifted. It's kind of like a relief that I personally could hardly imagine, but it would also bring up, I think, a little bit of a complicated feeling emotionally, because people have been shaped by HIV all their lives. Some of them, people have been born with HIV, and oftentimes people would think about, like Paul, friends who have passed before this cure has come about. And so I think a cure would ultimately be very powerful and very emotional, but it's going to be joy mixed with some grief, I think, and we have to be prepared for that.
Mm. So we just heard from Paul talking about his experience of a stem cell therapy. Obviously, he needed that for his cancer, but it
Mhm.
sounds pretty grueling. So when you talk to people living with HIV about a potential cure, what do you think their experience or expectation is for sort of clinical trials or treatments that are cell and gene therapies? What are they prepared to do to be cured?
Well, people are prepared to do everything. Because mostly when I talk about the trials, they realize that these trials are intense.
Mhm.
And so people go through things like leukapheresis. They have long hospital stays sometimes. Sometimes they even have chemotherapy and endless monitoring. There's also a sort of emotional stress they can go through because they're stopping ART, a lot of them, and they have to watch their viral load or wait for their viral load to reappear and go up. But most people say they're willing to do something that's really hard, but they don't want to do something that's going to be life-threatening. So they really want to make sure that the scientists are monitoring them and that they're being looked at very carefully, like because people are healthy on their meds and they don't want to gamble with their lives. So people will go very far because they want to help the community, they want to help the future generations. But that decision that they make ultimately is never simple, and it depends on each person's reactions to whatever the specific trial they're considering might offer.
Right. So then let me ask you specifically about gene therapy. Do do people have concerns about the safety of gene therapies or long-term effects, including, specifically about altering genes? We heard about CCR5. How do people feel about that?
Well, I think the biggest fear that people have is the unknown. We're talking about science fiction often when we talk about gene therapy. And so, what they understand, the minimal that they understand that, they can't take gene editing back. You can't unedit a cell if something goes wrong. And so, people worry about cancer risks that could come from that or those other sort of off target edits that they hear about. They also, some who get some knowledge, know that the immune system could overreact and they're worried about that. And sometimes they're just worried about something that might be unknown showing up 5 or 10 years after the therapy.
Yeah.
And so, it's a lot of science fiction, which leads to a lot of emotional fear, wondering, will I still be myself? Or what if the cure that I have causes something worse? So I think really what they want to know about is what are the long-term safety plans, and they want to also hear some honesty from researchers about what they don't know yet. I think that really sort of maybe not takes care of the fear, but really lets people know that they're talking with other human beings and not someone who's sort of an authority figure from a science fiction epic who's going to be doing experiments on them.
Yeah, and of course, as you know, in the whole field of cell and gene therapy, an important part of developing these treatments is sort of long-term follow-up of individuals. We hope and assume these treatments will be safe, but the first people to do this really have to be the pioneers, and they also graciously commit to a lot of follow-up after their therapy, a lot of blood draws and checking them to ensure that these things don't just work, but that they work safely. Yeah. Michael, do you have any concerns? We talk about cell and gene therapy, and at the moment, it's a very still a very expensive, fairly rare treatment. So, do you worry about access? If we did develop a cell and gene therapy cure, do you worry about access to that for everybody who would need it?
Yes, I think one of my biggest fears is that a gene therapy cure comes about, but it's only going to be for something that the wealthy people can reach. Only the rich people or the rich countries will get it. And I know that we've seen this with other treatments. First come the big breakthroughs that we have, but then the price tags follow along, and then the inequalities result from that, and privileged few. And we have to make sure that it reaches Black and brown people. We have to make sure that people in the South are getting it, low-income folks, people outside the major cities, and even people around the world. And so that's probably one of the biggest fears I have about this specific type of cure, is that access will be for everyone.
Right. So maybe the last thing I'll ask you, it's an open-ended question, but if you had one message that you could send to both the scientists working on this and the policymakers and politicians about cell and gene therapy for HIV, what would that message be?
I would like them all to really slow down and to listen to the people. Listen to the people, talk to the people if you haven't been talking with them. People are just not viral loads and cells. You're working with people who have full lives, and they have fears and hopes and families and futures. So we want to make sure that you're making the safety real, just not theoretical, and that you're making the access fair, and it's not just impossible. You want to be transparent about what you know and don't know, and never forget that the goal isn't just to cure a virus, but it's really to improve the lives of people who live with it. And that's what I would love them to really think about.
It's lovely. Thank you so much, Michael.
Thank you.
Right, so I think we're back to Christina now. She's going to lead us in a Q&A session. We've had some questions coming in from the audience.
Perfect. Thank you all so much. We'll get you pinned into the sort of Brady Bunch view for this Q&A. As a reminder, thank you to those attendees who have dropped questions into the Q&A. That is open. We'll continue to accept those as we go. But just to start, I wanted to ask about kind of Paula or Jonah, where is the science today? Paula, you gave a great overview of the kind of different cell and gene therapy approaches that are currently in play. So could you kind of talk about what stage are those research programs at? Are we, just doing the very early research? Have we moved into clinical trials? A little of both? What's going on?
So yeah, we have clinical trials in play for all of the therapies that I touched on. Again, only a handful of people at the moment. And some of that reflects that this is the way we do cell and gene therapies. They're still very new medicines, and we go slowly and carefully at the beginning. And also it reflects this idea that people living with HIV are often the pioneers in these types of treatments. Remember, I said that the first CAR T-cell trials were done in people living with HIV. In addition, we hear about CRISPR now as these genetic scissors, but there was an earlier version of genetic scissors called zinc finger nucleases that were also being used to target and cut out the CCR5 gene. And the first people who were treated with zinc finger nucleases were people living with HIV. So this community has a long history of kind of stepping up and being open to what are very experimental and first in human treatments. But the good news is that many of these therapies are being trialed in small clinical trials. And you bet if any of them work, that will be expanded. As you've heard from Paul and Michael, there's a thirst for these therapies.
Appreciate that. Another question in the chat, and this, Michael, you mentioned this kind of the question of access. So I just wanted to actually share ASGCT's perspective, which is that, access is a topic that we're very much paying attention to. We, strongly support efforts to make these complex living therapies more efficient to develop and to manufacture them, with the goal of really bringing down those development costs over time. Because as Michael said, these are really sort of first generation therapies still. And so those development costs are quite high. So I think number one, ASGCT, supports funding for robust research through NIH to develop things like improved capsids that can get into target cells more efficiently, or more precise gene editing technologies. And that does include through public-private partnerships, like the FNIH Bespoke Gene Therapy Consortium is working on, AAV, kind of a playbook for researchers to use. ASGCT is a proud founding member of that consortium. And then number two, ASGCT really promotes a responsive regulatory framework for these therapies that can reflect their unique characteristics compared to a small molecule or kind of a traditional drug. So that's things like, adopting innovative manufacturing solutions and the use of platform technologies, supporting convergence among global regulators to streamline development across borders, that companies aren't having to kind of reinvent the wheel with every new submission. So all of those things we think will bring down the cost to take these therapies from the bench to the clinic. And then I guess on the side of access to therapies once they receive FDA approval, we're very much supportive of efforts like the CMMI cell and gene therapy access model, which is an outcomes-based payment pilot that focuses initially on sickle cell disease, but could potentially expand out to other diseases. I think cancer, CAR T cell therapies for cancer has been tossed out as an option. And it's early days for that model, but we really think it has the potential to really improve access to sickle cell gene therapies in that Medicaid population. And if it expands, then that could really be transformative.
Yeah.
So that's all to say there are challenges. But there are lots of folks working.
Let me add on to that, Christina,
Please.
because you mentioned the cell and gene therapy access model for sickle cell disease. And I think this is something really important that previous and current administrations should be proud of. So just to recap, this is the idea that Medicare and Medicaid, that CMS can, if you like, oversee getting cell and gene therapy for sickle cell disease out to the communities that need it. And so this so-called access model, which is now being managed by Dr. Mehmet, this has enrolled, I think it's about 33 states, DC and Puerto Rico included, that are all now part of this consortium. And this, we're excited about this because it could be a model for how a future cell and gene therapy for HIV could also be managed. So the idea is that CMS, Centers for Medicaid and Medicare Services, will sort of oversee this. They will negotiate, hopefully, discounted prices from the manufacturers. They will peg payment to outcomes. So if you get a very expensive gene therapy and it doesn't work, I guess you maybe don't pay or don't pay as much. It's really based on having a successful outcome. And they'll also provide oversight and support and additional funding mechanisms to the states to help them bring a sickle cell disease gene therapy to their the people living with the disease in their state. So I'm excited by this because it really suggests this is the way of the future, that when we get effective curative cell and gene therapies for HIV, we're going to need an access model across the country to also make that available.
And I would say one thing, Paula, that you once said at one of our events that we did here in Seattle with you, what a beautiful problem to have to work on if we actually have a cure that works, to be able to figure out how to make it accessible to everyone. I mean, I think everyone would be excited. Knowing that there is a cure that works would be something that we would then be working our damnedest to make sure that we get that to be people and make it accessible to everyone. And I just really agree with that sentiment that you expressed a while ago. It keeps me going when I feel like sometimes that the access issue is insurmountable.
Sure. There are two more sort of technical questions in the chat that I'll bring up really quick. The first is, Paula or Jonah, I don't know if you'll know this off the top of your head, but what is the incidence of off-target impacts from current therapy candidates?
So, Jonah, do you want to take that, or I can chip in?
Go ahead, because I
Yes.
actually don't really know
Okay.
off the top of my head.
So again, it's an interesting issue, this. And with a lot of gene therapies, especially the ones that promise precision, like genetic scissors that are going after just HIV, or genetic scissors that are going after the CCR5 gene, because they offer precision, we possibly hold them to a really high standard of perfection, which is kind of at odds with how we do medicine. You know, when you take a drug for a disease, all drugs have off-target effects. We titrate the dose so that the off-target effects are trivial or manageable. And so I think we're starting to really look at gene therapy now, not through the lens of we have to have perfection, but through the lens of, will it work? And are any potential off-target effects within the realm of what could be manageable and accessible? Having said that, for sure, the technology is evolving every day, and the new reagents we have, including the scissors, are massively more precise, and our assays to look for off-target effects have also expanded, and the ways that we could think about managing or treating any off-target effects also are managing. So, and similarly, I would also add, and maybe Jonah can speak to this as well, that when we're talking about CAR T cells, or engineering the immune system to attack the HIV reservoir, do you remember the little seesaw that Jonah showed you? We also have to manage the fact that that is also potentially going to go a little bit after other tissues in the body. But again, there are drugs that we can use to kind of titrate everything down and keep a CAR T cell therapy or a graft versus reservoir therapy, as Jonah is working on, within this sort of happy balance of attacking and killing the HIV infected cells in the reservoir while mostly sparing the rest of the body. I don't know, do you want to add to that, Jonah? Hopefully I didn't completely mangle what you're doing.
No, that's exactly correct. Is thinking back to that seesaw, is that you absolutely, that's one of the things that we're trying to do, is make this as specific as possible. I tend to think of it as a dartboard where you want to hit at least within the colors, and then ideally the double bullseye or the bullseye, right? You want to get as close to that as possible. But as Paula rightfully says, there are always off-target effects to medications, and we just want to try and minimize that as much as possible. And that's what we're doing, is trying to refine, especially with this cellular approach with the graft versus reservoir, is to refine it so that you hit that target as closely as possible.
Mhm.
Appreciate that. There's another question in the chat about the newly proposed plausible mechanism pathway from FDA, whether it might improve investment from industry into promising cell and gene therapies, HIV or otherwise. I will say that's a very, very fresh announcement, so there aren't a lot of details in the public sphere about how that proposed program would work. But I can say that ASGCT is looking forward to learning more about what the full plan for implementation is. And in general, like I mentioned previously, we are in favor of regulatory flexibility that supports safe and effective therapies. So it's a bit of a non-answer until we learn more, but we're excited to find out what the plan is for that.
Yeah, and I would say from the point of view of people who are developing cell and gene therapies, we're kind of excited by things like the plausible mechanism pathway, because at the moment, what holds us up, if you like, is the massive amount of preclinical studies we have to do, and being able, and sometimes there's not a perfect set of experiments you can do, because what we're trying to do is so targeted, so precise, so specific, including for humans, which limits sometimes safety studies we could do in animals, because of all those criteria, which can be very problematic for developing cell and gene therapies. Having kind of a realistic set of expectations of how we could really start to assess whether a therapy is likely to be effective and is therefore worth the risk of putting it into patients is going to be very important. And maybe not with HIV, maybe our appetite for risk with HIV is a little lower, because we do have these amazing antiretroviral drugs. But when you have other diseases, devastating diseases where people have a very short life and suffer horribly. I think we all understand though that the risk benefit ratio is very, very different. So final thing I'll say is that the great thing about the field of cell and gene therapy is we learn from, all these different diseases learn from each other. Again, cancer and HIV, they walk hand in hand on many things, and the development of these therapies. And as Jonah said, the things we've learned from bone marrow transplants and graft versus leukemia, we're now trying to apply to graft versus HIV reservoir. So back and forth all the time. So anything that really helps the field of cell and gene therapy is especially going to help cell and gene therapy for an HIV cure.
Appreciate that. And I guess in our final couple of minutes here, I want to send this off kind of in a round for you. Are there any final thoughts that you want to leave the audience with? And maybe we'll start backwards, start with you, Michael, and then Paul, Jonah, and Paula.
I just really would love, for us to never forget that we're dealing with people, and that, we're not just trying to cure a virus, but we really want to improve the lives of people who live with this virus. And that should really be the goal. Whatever cures that we're coming up with are improving people's lives.
Yeah, and I would say that I really hope that funding for HIV will continue at the level that it had been at, and maybe even increase, because this is something that will help everyone in the world.
Definitely no argument from ASGCT on that one. NIH research funding is an absolutely critical component of everything we're talking about today. Jonah?
Yeah, I'd just like to end on the note that I kind of started with, which is that we really are at an exciting point for HIV cure. As you can, if you think back to that timeline that I showed of the individuals who've been cured of HIV, and you look at the number that's come out in the last couple of years, is really staggering. And if we continue down this pathway, I expect that those numbers will continue to grow. We'll learn more and more from each case, and it will sort of be a self-fulfilling prophecy where we'll continue to learn more to help us design better cures and curative approaches. Again, I really believe that we will cure HIV in my lifetime. And sort of to echo what Paul says, I hope that the funding continues and that this is something that we can claim, comes out of the United States of America.
And I'll end by, I guess, being a cheerleader for cell and gene therapy in general and HIV in particular. To me, the innovations, the progress we've made over recent years are just mind-blowing. This is going to become mundane. We are going to be able to cure so many currently uncurable diseases, including HIV. We've built this potential on the backs of huge amounts of research, a lot of it funded by NIH, basic research in unrelated fields, and it's all coming together. It's really, we're heading into a golden age for cell and gene therapy. Prices will come down because that's what always happens with innovation. And I really hope that we can, as Michael said, sort of keep our eye on accessibility and affordability as we do this. And I'm very excited that CMS is taking the lead on helping us do this, at least with sickle cell disease. So I'm very excited about the future for this field and for this disease.
Well, thank you to all of our panelists for sharing their expertise today. Thank you to the audience for being here. For your records, this briefing has been recorded, so it will be available on YouTube as soon as we can get it up and posted. If you'd like to learn more about anything that you've heard today, please do get in touch. We'd be happy to continue the conversation and really talk about the many ways that cell and gene therapies can potentially change the world. So thank you all so much again. We hope you had a good lunch hour, and cheerio.
Thank you.
Yeah.
Thank you.
Bye.
All right.